Good day, this is the Professor, CBMG PhD. Today's Wednesday, September 6, 2023. It is 8:34 and 13 seconds, p.m. Eastern Standard Time. And today's research, photosynthetic bacteria. So let's hop to it here. Enhancing cancer therapy using functionalized photosynthetic bacteria. Date August 29, 2023. Source: Japan Advanced Institute of Science and Technology. As selective targeting of cancerous cells poses major clinical challenges during cancer therapy. However, this limitation can be overcome by using bioengineered bacteria with highly optimized chemical modifications. A recent study demonstrates the use of chemically modified purple photosynthetic bacteria for the successful detection and elimination of colon cancer cells in a mouse model. The study also sheds light on the underlying mechanism of action. However, its efficacy may be hindered due to technical constraints and the potential development of antibiotic resistance. Hence, it's crucial to achieve the moderate yet effective chemical modification of bacteria for improved biocompatibility and functionality such that their medical abilities are not compromised. Recently, certain types of purple photosynthetic bacteria have come in the limelight for their potential to address the challenges of bacterial therapy. Exploring this further, reports the use of chemically modified purple synthetic bacteria for detecting and eliminating hard-to-eradicate cancerous cells in a mouse model. The scientific investigators and microbiologists selected Rhodopseudomonas palustris as the optimal bacterium for conducting the studies. Rhodopseudomonas palustris demonstrated excellent properties such as near-infrared fluorescence, photothermal conversion, and low cytotoxicity. It absorbs near-infrared light and produces free radicals, a property that can be utilized to kill cancer cells. In an attempt to improve the therapeutic efficacy of the isolated strain, the team sought chemical modifications to alter the bacterial membranes. First, they performed membrane PE glycation or the attachment of polyethylene glycol derivatives to the bacterial cell walls. And prior research indicates that bacterial PE glycation helps in evading host immune response and converts light into heat energy, which can then be utilized to selectively eliminate cancerous cells. The initial results were encouraging. For instance, coating the Rhodopseudomonas palustris membrane surface with a biocompatible anchor or membrane did not adversely affect Rhodopseudomonas palustris cell viability for at least a week. Moreover, the biocompatible anchor or membrane functionalized Rhodopseudomonas palustris was not eliminated via phagocytosis by macrophages, cells that play a key role in the immune system's defense actions against bacterial invasions. Next, the researchers attached a fluorescent Elixir 488 BSA conjugate to the biocompatible anchor for membrane functionalized Rhodopseudomonas palustris, thus creating a bacterial complex with a trackable fluorescent marker. And this conjugate was subsequently replaced with PD-L1 antibody. Prior studies have shown that cancer cells express a protein called programmed cell death ligand 1 PD-L1 on their surface. PD-L1 can smoothly turn off the host defense system by binding PD-1 receptors. And this allows the cancer cells to evade the immune detection and elimination. Anti-PD-L1 antibodies block this interaction, thus preventing cancer cells from bypassing immune system-mediated destruction. As expected, both anti-PD-L1 biocompatibility anchor for membrane Rhodopseudomonas palustris and Rhodopseudomonas palustris inhibited tumor growth in a marine model of colon cancer. However, anti-PD-L1 biocompatibility anchor for membrane Rhodopseudomonas palustris and biocompatibility anchor for membrane Rhodopseudomonas palustris and Rhodopseudomonas palustris, when excited with a laser, showed an especially dramatic anti-cancer effect In fact, solid tumors vanish completely following the laser irradiation of anti-PD-L1 biocompatibility anchor for membrane Rhodopseudomonas palustris, biocompatibility anchor for membrane Rhodopseudomonas palustris, or Rhodopseudomonas palustris that were injected into tumor-bearing mice. And further, on assessing photothermal conversion properties, both anti-PD-L1 biocompatibility anchor for membrane Rhodopseudomonas palustris and natural Rhodopseudomonas palustris exhibited strong photothermal conversion due to the presence of light-driven bacterial chlorophyll molecules. Among the various bioconjugates, anti-PD-L1 biocompatibility anchor for membrane Rhodopseudomonas palustris showed the highest efficacy in the initial stage of the treatment. Moreover, it was non-toxic to surrounding healthy cells or to the marine host. Subsequent experiments revealed the underlying mechanisms of colon tumor annihilation in the mouse model. The findings revealed that light-driven functional bacteria demonstrated effective optical and immunological functions in the marine model of colon cancer. Moreover, the near-infrared fluorescence of the engineered bacterial complexes was used to locate tumors effectively, paving the way for future clinical translation. The scientific investigators and microbiologists believe that this bacterial technology could be available for clinical trials in 10 years and have positive implications for cancer diagnosis and therapy, hoping that bacterial therapy helps researchers, oncologists, and patients with cancer with much needed relief. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Teacher of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.